In this video, we examine the approach needed in statistical inference to study the association between two categorical variables. First, a reminder. When we have categorical data, what we record for each individual is some feature, some characteristic, and we can summarize this variable either by looking at the frequency distribution. That means computing the count of individuals that have each of the characteristics that make up the variable, or from a relative frequency perspective, calculating the proportion of individuals that have each outcome. We also discussed the concept of independence. Two events are independent if the knowledge that one event is true or has happened does not affect the probability of the other event. We also described the multiplication rule for independent events, which states that if we have two events A and B that are independent, then the probability of A and B happening together jointly is simply the product of the two simple probabilities, probability of A times probability of B. So let's now see how this relates to the association between two categorical variables. One way to summarize the association between two categorical variables is to represent the findings in a two-way table that uses the rows for all the outcomes of the first variable and the columns for all the outcomes that make up the second variable. And then inside the cells of that two-way table, we can have the counts of individuals that have the two related features. We saw that probabilities can be computed very easily from such a format. For example, the probability that a randomly selected individual would have both the A and B features can be computed as the number of individuals that have both A and B features, the cell count, divided by the table total, the number of individuals in the entire data set. The multiplication rule for independent events would then translate into the cell count for the A and B cell is equal to the row total for outcome A times the column total for outcome B divided by the table total. So let's now put this in the perspective of statistical inference. If the data were obtained on all the individuals in the target population, then this equation would be exactly correct when the two events are independent. If instead the table of counts is based on only sample data, then we need to account for the fact that random samples vary, and therefore we will need to test a hypothesis about the properties of the population using the sample data as evidence. We have seen that when we want to test a hypothesis about some property of a population, we start with a null hypothesis, which is typically a statement of no effect, no difference, nothing special going on about the target population. We then use the sample data to calculate a test statistic and a test p-value that tell us to what extent the sample data deviate from what we would expect if the null hypothesis was true. So consider a situation where we have collected data on a sample of individuals such that we have two categorical variables that can be summarized in a two-way table with R different rows and C different columns. We will test the null hypothesis that there is no association between the two variables used to make this table, the row variable and the column variable. That means those two variables are independent in the population from which the data were taken. Our alternative hypothesis is going to be non-directional. We are simply saying that the null hypothesis is not true. There is some association between those two variables. They are not independent in the population. So we need to find a way to compare the sample data to what we would expect if the null hypothesis was true. In this case, when the null hypothesis is true, we would expect each cell in the two-way table to be equal to the row total times the column total divided by the table total. And we will use those expected counts to calculate a test statistic that will tell us how much the sample data deviate from what we would expect if the null hypothesis was true, relative to how much variability can be expected just based on the random sampling process. So in this case, the test statistic is called the chi-square statistic, and it is calculated as a sum of components with one component for each cell in the table, computed as the observed count minus the expected count square divided by the expected count. Mathematically, it can be proven that when the null hypothesis is true, the chi-square statistic follows a skewed distribution called a chi-square distribution, specifically the chi-square distribution identified by its degrees of freedom equal to number of rows minus 1 times number of columns minus 1. So since we have a probability distribution for the test statistic when the null hypothesis is true, we can calculate the test p-value, which is the probability when the null hypothesis is true, of obtaining a test statistic at least as extreme as that computed, so the right tail of this skewed distribution. And when we have this test p-value, we will use it as we have before. A p-value that is not particularly small indicates that the data are not inconsistent with the null hypothesis. Therefore, the findings are inconclusive and we can only fail to reject the null hypothesis. On the other hand, if the p-value is small, it provides some evidence against the null hypothesis, and smaller p-values provide stronger evidence against the null hypothesis and in support of the alternative. For your information, here is what chi-square distributions look like. The different degrees of freedom just indicate where the peak of that distribution lies and how skewed that distribution is.